y'all, my name is Parul, and I will be doing nucleic acids as the inheritable informational molecules, aka DNA. So a nucleic acid are molecules that carry hereditary genetic information of the organism. A monomer of the nucleic acids are nucleotides, which consist of a 5-carbon sugar, a phosphate group, and a nitrogenous base. They're inheritable since the genetic information is passed from generation to generation using gametes. Two examples of these are DNA and RNA. So the DNA structure has a double-stranded helix, and the polynucleotides are the helix with the bases extending into the center, which is shown in the picture. And the DNA is wrapped around histones. The latter, as you see, are alternating phosphate groups and deoxyribose, which is a sugar. And the double-stranded helix is antiparallel, meaning one side goes from a 3' to a 5' direction, and the other goes to a 5', 3' direction. You can tell which is the 5' direction from the phosphate, so just remember 5-phosphate. And so another way to look at the antiparallel is the sugar and the phosphate are running in opposite directions. One kind of looks upside down. So some of the bases are ATGC, which are the base pairings, adenine and thymine, and guanine and cytosine. And thymine and cytosine are pyrimidines, and adenine and guanine are purines. And these are two types of nucleotides. And the pyrimidine hydrogen bond to the purines to create the rungs of the DNA ladder, so therefore A and T are hydrogen bonded, and so are G and C. And RNA, there's 3 types of RNA. There's messenger, transfer, and ribosomal. It's single-stranded, and instead of thymine, RNA uses uracil, so it's AU and GC. So the replication process for DNA goes into the 5', 3' direction, and helicase first unzips the DNA into 2 antiparallel strands. After that, the DNA polymerase III latches on at one end of the opening, called the replication bubble, which is on the 5' end, and begins to continuously lay new DNA strands from free nucleotides in the nucleus. And helicase continues to unzip, so the process continues without interruption. And on the other strand, it's not that easy. It's called the lagging strand. The RNA primase lays short segments of RNA primer nucleotides at as many points along the strand. And then the polymerase III, DNA polymerase III, fills in between the RNA primers with DNA nucleotides. After that, the DNA polymerase replaces the RNA primer with DNA, and these segments of DNA are called Okazaki fragments. After that, the DNA ligase comes in, an enzyme, and joins the DNA fragments together, and you get 2 identical strands. Woo! Okay, and other stuff about replication. In eukaryotic chromosomes, replication is initiated at many points, and DNA is semi-conservative, meaning it keeps half of its original old DNA, and then it has half of new DNA, which could be represented by the 2 colors I used in the other slides. So transcription, it's a synthesis of a strand of RNA using a strand of DNA as a template, and transcription is also carried out in a 5' and 3' direction. Reverse transcriptase, catalyzes the production of DNA from RNA. And so the process is an enzyme where RNA polymerase moves along the DNA strand, temporarily unwinding the double strands. The nucleotides attach to the template strand and elongate the forming RNA strand. The building RNA strand is separated from the DNA as the enzyme moves along the DNA and the DNA rewinds. RNA differs from DNA in composition, where all instances of thymine are replaced by uracil. So basically, the RNA is, the whole point of transcription is to produce messenger RNA, which is going to be used in translation later on, which is what this is. Translation, this is basically a small overview of it. It has 3 steps: initiation, elongation, and termination, and it folds into a 3D structure eventually after it goes through the cycle. So a codon is a sequence of 3 nucleotides which together form a unit of genetic code in a DNA or RNA molecule. And the steps to initiation is first the mRNA is bound to ribosome with the start codon at a P site. At transfer RNA molecule, with the amino acid methionine and the anticodon UAC, has bounded to the exposed start codon right here. And the second RNA molecule with the anticodon GCA and the amino acid serine has bounded to the A site. The two amino acids are close enough to form a peptide bond between them. A peptide bond is formed between the methionine and serine, and the peptide is bound to the site. And the methionine tRNA leaves, and the P site is now exposed. So this process continues. The three sites are EPA, which is how the process keeps going. And after this process continues, it's elongation. The ribosome then moves one codon down to the mRNA in a 5' 3' direction, and this continues going on until there's a polypeptide chain. So the termination process happens and when the ribosome reaches a stop codon on the mRNA. While there's no transfer RNA for a stop codon, there's an enzyme called the release factor, which cuts the polypeptide chain, resulting in a new protein when it develops into a 3D structure. The ribosome separates, and the mRNA is released to be used again or degraded. And translation occurs at multiple sites along the mRNA. So these are some links I used, and thank you very much, guys. Hope you guys get those 5s and 7s on the AP and IB exams. If you have any questions, go ahead and Facebook stalk me, and good luck. Yay!